Right guys, we have got phloem tissue here. Phloem tissue, what it does is it transports solutes. It is an energy requiring process down here or up here if you want to read it. Basically what it does is it carries the solutes from wherever it's produced to wherever it's needed. There are two important cell types you need to know. You've got sieve tube elements and you've got companion cells. I wrote this 1-to-1 situation over here, this ratio, to say basically that for every sieve tube element you will need a companion cell. So you've got the sieve tube element here, this clear sort of thing here, and then you've got your companion cell with the nucleus and all the different organelles as the black dots there. So the reason you actually need a companion cell is because the sieve tube element has no nucleus, as you can see in the diagram, and it's got few organelles, so there's literally nothing inside it, and that means it can't carry out living processes such as providing energy. It's got no mitochondrion. So that's why the companion cell is needed. So the companion cell provides energy for active transport of solutes. Now, moving on, that's all we need to know about sieve tube elements and companion cells. 1-to-1 ratio, no nucleus, few organelles, and that the companion cell is needed to provide energy. Done. Translocation. So translocation is the movement of solutes. So basically, that's phloem. Translocation is the movement of solutes, and it's an energy requiring process. So when phloem moves the solutes, it needs energy to do so. So this is when we come to the source and the sink. So the source is where everything is produced, such as the sucrose like this, mainly found in, that's the main substance you'll find, which is produced by the leaves when photosynthesizing. So sucrose is the main substance produced, but it will produce other substances as well. So that's at the source, and there'll be a high concentration of sucrose, or substances, and there'll be a low concentration down at the sink. So the sink is where everything is needed. So places like the stem, the roots, so meristems basically, anywhere that's got growth or is growing. Down at the sink you will find enzymes. Now enzymes are found here because what they do is when the sucrose reaches this sink and is still being produced at the source, the enzymes will break down the sucrose down here into something else, or they'll turn it into starch or something like that, so that it maintains a concentration gradient. So there'll be always a low concentration gradient here and a high concentration gradient. We know that substances move down the concentration gradient from high to low. So that's what we really need to know about phloem tissue with that. And now we're going to move on to the mass flow hypothesis. Right, so in the mass flow hypothesis, basically it's understanding why things move down, why things move from the source to the sink. So we've got active transport from the of the solutes going from the companion cell into the sieve tube element into the actual main vessel. Now, why they do this is because there's all of this solutes is being produced further down, which I will explain in another video, and it will come into the companion cell, come into here, so there'll be like other cells here joined up along here, move this way. Now this will have a high concentration gradient, but this will also have a high concentration gradient already. So the companion cell moves all the stuff in by active transport, and then this creates a low water potential, because if you've got high solutes, high amount of substances, it's going to be a high concentration, so there'll be a low water potential. So that means water will move in at the source via osmosis, or diffuses in via osmosis from the xylem vessels or the companion cell, so water will move into the phloem vessel. Now this will increase the pressure. Straight up, it will just increase the pressure. Now at the sink end, solutes are removed, like I said earlier, by the enzymes that are either stored as starched or used. Used meaning they're being broken down by the enzymes to be used in respiration down here. So this will increase the water potential because all the solutes are being used, which means there will be a low concentration of solutes and a high water potential. So that would decrease the pressure. So notice that we've got an increase in pressure at the source and a decrease in pressure at the sink. So that means a pressure gradient is formed. That's a key word there. Pressure gradient is formally for like a 4 marker that will be needed, and this will push the solutes down because of this pressure gradient. So like this, the high pressure here and the low pressure here, solutes will move down from the source to the sink, and then they'll be broken down, and it's almost like a cycle, happen again and again and again. When the companion cell brings in all the solutes via active transport, pressure will be high because water will enter because of the concentration, everything will move down. And it just happens, it goes on and on. So when the solutes are down here, the sucrose that will be used in respiration or stored, such as starch. Thank you for watching. Like and subscribe. That is phloem. Cheers.